In the 4th century, a Chinese physician wrote down a prescription for intermittent fevers. He was specific. Take the fresh plant, soak it in cold water, wring out the juice, and drink it. Not boiled, not heated, cold. 1700 years later, that detail, overlooked by every Western researcher who had tested the plant and found nothing, would prove to be the key to isolating one of the most important antimalarial compounds ever discovered. A compound that has since saved hundreds of millions of lives. A compound that would win the Nobel Prize in medicine. And it came from a plant that most botanists dismissed as a common weed. Malaria is ancient. It has stalked humanity since before we were human. Parasites in the blood, transmitted by mosquitoes, causing cycles of fever, chills, and death that have shaped the course of civilizations. Empires have fallen to it. Wars have been decided by it. For most of human history, there was no reliable cure. By the 20th century, modern medicine believed it had the answer. Quinine, extracted from the bark of the South American cinchona tree, had been the standard treatment for centuries. Synthetic alternatives followed. Chloroquine, developed in the 1930s, became the frontline defense. It was cheap, effective, and widely distributed. And then the parasites adapted. By the 1960s, drug-resistant malaria had emerged in Southeast Asia. Chloroquine was failing. Soldiers were dying, not from combat, but from a disease that medicine could no longer control. The crisis was acute in the jungles of Vietnam, where both sides of the conflict faced an enemy that no weapon could defeat. Pharmaceutical companies searched for new compounds. Laboratories tested thousands of molecules. Progress was slow. Resistance spread faster than solutions could be developed. And yet, the answer had already been written down. It had been written in the 4th century, in a text compiled by a physician named Ge Hong. The book was called Emergency Formulas to Keep Up One's Sleeve, a practical manual of remedies for conditions that travelers might encounter far from home. Among its entries was a treatment for what the text called intermittent fevers. The prescription was precise. Take a handful of the fresh herb, soak it in cold water, wring out the juice, drink it, not boiled, not prepared as a traditional decoction. Cold. This detail mattered more than anyone would understand for another 17 centuries. The plant described in that ancient text would eventually yield a compound more effective against drug-resistant malaria than anything modern chemistry had produced. A compound that acts faster than quinine. A compound that attacks the parasite at multiple stages of its life cycle. A compound that has since been administered to hundreds of millions of patients in some of the poorest and most vulnerable regions on Earth. The discovery of that compound would earn its principal researcher the Nobel Prize in Physiology or Medicine in 2015. But the knowledge was always there, written down, preserved, waiting for someone to read it correctly. The plant has been used in traditional Chinese medicine for at least 2,000 years. Its Chinese name, Qinghao, appears in some of the oldest pharmacological texts in existence. The Shenong Ben Chao Jing, a foundational text of Chinese herbalism compiled during the Han Dynasty, lists it among hundreds of medicinal plants. It was known even then as a treatment for fevers, though the specific mechanisms were, of course, not understood in modern terms. What traditional practitioners understood was that the plant worked. It reduced fevers. It seemed to break the cycles of chills and sweats that characterized certain illnesses. It was bitter, aromatic, and widely available, a common plant that grew readily in temperate regions across China and beyond. But it was Ge Hong's 4th century text that preserved the critical detail. Ge Hong was not a court physician. He was a scholar practitioner who lived during a turbulent period of Chinese history, a time of political fragmentation and constant warfare. His manual was designed for practical use, for travelers, for soldiers, for people who might find themselves far from medical help and in urgent need of treatment. His instruction for intermittent fevers was unusual. Most traditional preparations involved heating, boiling, creating decoctions that concentrated the active compounds through prolonged cooking. But Ge Hong specified cold water extraction. He specified fresh plant material. He specified wringing out the juice. He did not explain why. Perhaps he did not know why. Perhaps he had simply observed that this method worked when others did not. Traditional medicine often preserves practical knowledge without theoretical justification. The knowledge that something works, passed down through generations, even when the reason remains unexplained. For 1700 years, this prescription existed in the historical record. Scholars studied Geihong's text. Herbalists continued to use the plant. The knowledge was not lost. But it was not found either, not by the researchers who would eventually need it most. When Western scientists first investigated Chinese medicinal plants in the 19th and early 20th centuries, they followed their own methods. They prepared extracts using heat and alcohol. They applied the techniques of European pharmacology to materials they did not fully understand. They tested this plant. They found nothing remarkable. The active compound, it would later be discovered, is destroyed by high temperatures. The very methods that Western science considered rigorous were eliminating the therapeutic value before it could be measured. The plant was tested and dismissed. The knowledge in Geihong's text, the specific instruction to use cold water, was overlooked or ignored. The plant was categorized as a minor herb. Interesting historically, perhaps, but of no practical value to modern medicine. This conclusion would persist for decades. By the late 1960s, the situation had become desperate. Drug-resistant malaria was spreading beyond Southeast Asia. The global health implications were staggering. Malaria kills primarily in the tropics, in sub-Saharan Africa, in South Asia, in regions where healthcare infrastructure is fragile and resources are scarce. A disease that resisted all available treatments threatened millions of lives. In 1967, the Chinese government launched a secret research initiative. The project was classified, given only a code designation derived from its start date, May 23, 1967. The goal was to find new antimalarial compounds by any means necessary, including the systematic investigation of traditional Chinese medicine. Hundreds of researchers were assigned to the project. They reviewed ancient texts. They collected plant samples. They tested thousands of preparations using modern laboratory methods. Among the researchers was a pharmaceutical chemist, trained in both Western and traditional Chinese medicine. She was assigned to review historical records, searching for any mention of fever treatments that might contain overlooked compounds. She found Geihong's text. She noted the unusual instruction: cold water extraction of fresh plant material. She recognized that previous researchers had used heat-based methods. She wondered if that was why the plant had shown no activity in earlier tests. She changed the approach. Using low-temperature extraction methods, techniques that preserved the heat-sensitive compounds, she prepared new extracts from the plant. She tested them against malaria parasites in the laboratory. The results were dramatic. The extract showed powerful antimalarial activity. Further refinement isolated the specific compound responsible. By 1972, the active molecule had been identified: a sesquiterpene lactone with a chemical structure unlike any existing antimalarial drug. The compound worked differently from quinine and its derivatives. It contained an unusual peroxide bridge that reacted with iron in the parasite's digestive system, generating free radicals that killed the organism rapidly. It acted faster than any previous treatment. It was effective against strains that had developed resistance to chloroquine. But the political situation in China during the 1970s made international communication difficult. The research remained classified for years. The findings were not published in Western scientific journals until the 1980s. The compound did not receive widespread international attention until the 1990s. By then, drug-resistant malaria had become a global emergency, and the solution, discovered 2 decades earlier, derived from a 4th century prescription, extracted from a plant that Western science had dismissed, was finally recognized. The World Health Organization now recommends treatments based on this compound as the first-line therapy for uncomplicated malaria. Combination therapies pairing it with other drugs have become standard protocol across Africa and Asia. The plant did not fail. Western science had simply failed to read the original instructions correctly. The plant that produces this compound is, by most measures, unremarkable. It is an annual herb, completing its life cycle in a single growing season. It reaches heights of 1 to 2 meters, with feathery, deeply divided leaves and small, inconspicuous yellowish flowers. It is aromatic, strongly scented when crushed, with a bitter taste that discourages grazing animals. It grows readily in temperate and subtropical climates. It tolerates poor soils. It requires moderate water but survives periods of drought. It is naturally pest-resistant, thanks to the same bitter compounds that make it medicinally valuable. Insects avoid it. This makes it useful as a companion plant and as a natural pest deterrent in gardens and fields. From an agricultural perspective, it is a low-input crop. It does not require heavy fertilization. It does not demand pesticides or herbicides. It can be grown in marginal land that would not support more demanding crops. In several developing countries, it has become a cash crop for smallholder farmers, a source of income from land that might otherwise produce little. The compound content varies depending on growing conditions, harvest timing, and genetic variety. Plants grown in certain regions under specific conditions produce higher concentrations. This variability has been both a challenge and an opportunity. A challenge for pharmaceutical standardization, an opportunity for agricultural development in communities that need economic alternatives. Modern cultivation programs have worked to develop higher-yielding varieties. The compound can now also be produced through synthetic biology, engineered yeast cells that manufacture the precursor molecule, which is then chemically converted to the final product. This has helped stabilize global supply, reducing dependence on agricultural production alone. But the plant itself remains in cultivation. It is grown in China, Vietnam, East Africa, and elsewhere. The market fluctuates. As synthetic production increases, agricultural prices sometimes fall, but the crop persists. Farmers who have grown it for decades continue to do so. The plant has also found secondary uses. Its essential oils are studied for antimicrobial properties. Its traditional use as a fever remedy continues in some regions, though health authorities caution against substituting raw plant material for standardized pharmaceutical preparations. The compound works best at specific doses, in combination with other drugs, administered according to protocols designed to prevent the development of resistance. The ecology of the plant reflects its resilience. It self-seeds readily. It colonizes disturbed ground. It is considered invasive in some regions, spreading beyond cultivation into roadsides and waste areas. What makes a plant a weed in one context makes it a survivor in another. This is a plant shaped by its chemistry, bitter, aromatic, avoided by pests, toxic to parasites. These same qualities made it medicinally valuable to practitioners who observed its effects without understanding its molecules. The knowledge was empirical. It was practical. And it was correct. The plant is called sweet wormwood. In Chinese, it is Qinghao, a name that appears in texts spanning 2 millennia. In scientific literature, it carries a Latin binomial that places it within the Artemisia genus, a group of aromatic plants that includes common sagebrush and culinary tarragon. The compound extracted from it is called artemisinin. The researcher who led its discovery was Tu Youyou, a pharmaceutical chemist born in 1930 in Ningbo, China. She spent her career at the China Academy of Chinese Medical Sciences, bridging the traditions of Chinese herbalism and modern pharmacology. In 2015, at the age of 84, she was awarded the Nobel Prize in Physiology or Medicine for her discovery, the first Chinese Nobel laureate in that category, and the first to win for work derived directly from traditional Chinese medicine. Her achievement was not merely chemical, it was interpretive. She read the ancient texts with the eyes of a trained scientist, recognized that earlier researchers had used the wrong methods, and had the insight to change the extraction process. She understood that Ge Hong's instruction to use cold water was not arbitrary. It was essential. Today, artemisinin-based combination therapies are the standard of care for malaria across most of the world. The World Health Organization estimates that these treatments have saved millions of lives since their widespread adoption. In regions where malaria was once a leading cause of death, mortality rates have fallen dramatically. Sweet wormwood can still be found growing in temperate Asia, in parts of Europe, and in regions of North America where it has naturalized. It is cultivated commercially in China, Vietnam, Tanzania, and Kenya, among other countries. Smallholder farmers grow it as a cash crop. Pharmaceutical companies contract for standardized harvests. Researchers continue to study its chemistry and ecology, For those who wish to grow it, the plant is not difficult. It prefers full sun and well-drained soil. It tolerates drought once established. Seeds can be started indoors and transplanted after frost, or sown directly in warm soil. It grows quickly, reaching full size within a few months. The leaves are most potent just before flowering. Traditional harvesters knew this, timing their collection to maximize therapeutic value. The plant is bitter. It is aromatic. It is, in most respects, a common weed, the kind of plant that gardeners might pull without a second thought, that grows in disturbed ground and forgotten corners. But within its tissues, synthesized through pathways that evolution shaped over millions of years, is a molecule that has changed the course of global health. A molecule that was described in a 4th century prescription. A molecule that Western science overlooked for decades because it could not imagine that ancient physicians had already found the answer. The knowledge was always there. It was written in texts that scholars had studied for centuries. It was preserved by traditions that modern medicine dismissed as primitive. It was encoded in a simple instruction: use cold water, not heat, that proved to be the difference between failure and success. Tu Youyou read the old texts. She followed the old instructions. And she found what generations of researchers had missed. The plant still grows. The compound still saves lives. The lesson endures. Sometimes the most important discoveries are not inventions. They are remembrances.